Great, thank you. Yeah, so I'm Lydia McGill. That was a pretty good introduction, so I don't think I need to tell you any more about myself. I'll just jump right on into it. So I wanted to just start with a little poll. So do you know what flame shells are, basically, just to see what people know about the topic? So hopefully that poll's come up on your screens now. If you just want to fill that in. And don't be afraid to say no if you know nothing about flame shells, because most people I talk to have never even heard of them. So yeah, I won't be surprised. Okay. So most people,
we're going to end that.
Yeah, cool. It looks like most people do have a little bit of knowledge, which is great. Some people not really knowing what they are. And yeah, a good number of people researching them, which is really good. Like that. Okay, so I'll just go into a little bit of background for those who don't know. Here we go. Okay, so flame shells are a bivalve mollusk which can be found across the Atlantic coast of Europe, mostly at depths of around 5 to 30 meters. Large populations of flame shells are found on the west coast of Scotland. They tend to be found in areas with strong currents, so the west coast is very suitable for them. And many of these populations are found in sea lochs. The largest known population in the world is located in Loch Carron, which was just found back in 2017. So we're still finding more of these, and especially now, I think there is a growing interest in them, and we seem to be stumbling across them, with populations now being found in Ireland, and just recently a new population was found off the coast of Arran. So it's all quite exciting stuff. So why are we getting more interested and studying flame shells? So as well as being of interest just due to the large numbers of the species that we find in Scotland, the species is of high ecological importance. So flame shells create nests using byssal threads, which they produce and use to bring together bits of debris found on the seafloor. I actually found this picture in NatureScot's pictures the other day. So you can actually see threads coming out and around the nest, holding it all together. So I thought that was really cool, and that I'd share that with everyone. So yeah, these nests start to combine and form a dense bed, which raises and stabilizes the seabed, making a habitat that is more suited to hundreds of other species as well. But these beds are vulnerable to mechanical disturbance, so say bottom trawling or dredging, which bring up the bed and leave like scars, which then need to heal. This we know happened to the bed in Loch Carron not long after its discovery, and this led to an emergency marine protected area being put into place to protect. So we know these disturbances are damaging, but we don't know much about bed recovery, and this is quite important for us to understand for management. So my internship at NatureScot was 3 months long. I did it at the end of 2020. So of course it was a working from home situation, probably pretty different from normal internship, but it still worked pretty well. I'm happy with how it went. The data I was using for the internship was collected previously, with NatureScot collecting sediment samples from various flame shell beds and identifying the species present in those samples. So this data was just waiting to be analysed, and that's where I come in. So the 4 lochs involved in this study can be seen in the map here, with 2 beds being sampled from Loch Carron to bring the total number of beds sampled to 5. 2 different sampling methods were used when collecting samples, so grab sampling and core sampling. So these differ, as grab samples are 4.5 times greater in volume than core samples. So this is something we must take into consideration when we compare the different beds. Samples were also collected over different years, which again could affect the results of the study. And this is another thing which we must consider when analysing results and comparing beds. and depth of the beds was also considered as a potential factor affecting results. So what did we want to find? So we wanted to analyse the species composition of the beds, but also to try functional trait analysis, which I'll explain a little more before I go into those results. But our main interests were finding if there were any differences between the flame shell beds. For this, I had to consider different factors such as location, year, sampling method, or bed depth. It was also interesting to look at the results in the context of management, looking at how long protection was in place before sampling was carried out at different locations. And finally, just to learn more about the beds in general, since we don't currently have a lot of knowledge on flame shell beds. So with the species data, I used non-metric multi-dimensional scaling and analysis of similarities, which measures the difference between the beds using a species by abundance matrix. I also used various diversity indices to further compare beds and performed likelihood ratio tests to test the significance that various factors such as location or year had on the results. So Shannon's index accounts for both abundance and evenness of the species present. Pailou's evenness looks at the species diversity along with species richness. Richness is average number of species within the samples in the area, and abundance is average number of individuals within the samples in the area. So the NMDS plot to the left shows us that each location is quite distinct, with the two sample groups from the Lochfine group at the top, the two sample groups from the two separate beds in Loch Crieran to the bottom right, and Loch Broom and Loch Carron clustering to the left, but still distinct from one another. This appears it could be more clustering by location, but these clusters also correspond to the years in which sample collection took place. So we can see 2015 at the top, 2017 over here, and 2012 to the right. The Lochfine clustering is promising as it shows that there is a lot of similarity in the two sampling methods used here, because that one is grab and one is core. The Anderson to the left shows that overall the most difference is found between beds rather than within. But we do see quite a lot of differentiation within both Lochfine sample groups, which is very different from the other beds. This bed was actually sampled before protection was put into place. So this is quite interesting that it would come up like this. We also see both Loch Crieran beds have the least differentiation between samples within, which is an interesting contrast to Lochfine findings, as Loch Crieran was protected for 5 years prior to sampling. So these results seem to hint that maybe with the protection in place, the beds are reaching some kind of equilibrium in what species are present, as the samples are differing less the more time in which the beds are protected. So moving on to the diversity indices, we noticed that the core samples, which are the bottom three, have much lower abundance than the grab samples, which is the top three. This makes the value difficult for us to compare across these methods. I did try adjusting this, so core samples were 4.5 times smaller in volume than the grab samples, but adjusting the data to match the volumes just made the abundance values for core data disproportionately higher than that of the grab samples instead, so it just kind of reversed things. So with the other indices, luckily they didn't take abundance into account, so they are a little more comparable across the methods used. It's interesting that we see high species diversity and richness in Loch Carron when this bed was damaged not long before the samples were collected. This could be due to a new species moving into the damaged area, which may then level back out again after some time. Despite this high diversity and richness, in comparison with the other grab samples, Loch Carron has a low abundance. It would be interesting to sample this bed again and repeat these measures to compare the values found now after a few years of recovery to those values that were found immediately after the damage took place. And then the likelihood ratio tests. So here I tested the effects of location year on the diversity measures, as well as depth and sampling method, but tests for depth and sampling method did not find any significant results, so these are not shown here. Location was found to significantly correlate with the diversity index, species richness and abundance, while the year was significant for diversity index and species richness. So these results suggest that there may be fluctuations in species present over different years, but also that species may just differ in the different locations where the beds are located. So it would be useful to sample all locations in the same year to clarify this. So for the functional traits analysis, this meant that I had to assign the traits of each species with a number to represent their affinity to each trait. So the traits were divided into categories. So for example, bioturbation included biodiffuser, surface modifier, and other traits which species would fall into. The species traits were identified using biological trait databases such as polytraits or worms. So this method was adapted from another NatureScot report in which the method was used to compare different habitats, which I don't believe has been published yet. So the purpose of these analyses were to see if, despite species differences, beds were functionally similar. So although different species were present, were they fulfilling the same functional roles? I did these analyses for abundance data where the scores for each species were multiplied by the amount of the species present in each sample, and also for proportion data where these values were all altered to be of equal proportion. So this would minimize effects of dominant species or traits, so we can have a closer look at the finer scale differences. So for the abundance data first, here to the left, we see that the PCA, where most of the beds are completely clustered together in the middle, with just 2 sample points drawing out, one for Loch Carron to the top and one for Loch Fyne down to the bottom left. So Loch Carron's area of differentiation could be linked with the damage, but we're not sure what the Loch Fyne sample point could be different for what reason. And over to the right is a figure showing the directions of traits in correspondence with the PCA. So we see that the reason the Loch Fyne data point is so drawn out is because there are a larger number of species with the surface modifier trait. So the Loch Carron point appears to have more suspension and deposit feeders. It would have been quite interesting if detritus feeding as a trait had come up here correlating with the Loch Carron point of differentiation. So that would maybe help us link it with any damage or anything, but we don't see this coming up. So it's interesting as to why it's different. And then with the proportion data, we see less of an influence of those dominant traits. So we can see those points kind of closer up, and we see there actually isn't a complete overlap across all of the beds. There appears to be some gradual differentiation across the beds, but surprisingly we see that one of the Loch Carron beds is very different from the others, which is interesting and seems to contrast with what we found in the species analyses. So in summary for that, species analyses seem to indicate that variations in the species present in beds could be influenced by location and by year, and that the species present in the beds do differ. These analyses also hint at differences within beds linked with the length of time in which beds are protected. And then with the functional trait analysis, we see that although the species are different, they do appear to be fitting the same ecological niche. So this would suggest that the beds are not so different across locations or year, but only in the species present. And I believe that these analyses could be a good basis for future monitoring. So to look into this further, I suggest repeating sampling in the future, but this time collecting samples in the same year using the same method for all the collections, just to ensure comparability of all of the results. To repeat these analyses with a more comparable data set would help us understand what exactly we can learn from this data. And this could help us to build on our knowledge of flame shell beds, and repeating the collections over the years could be more informative to see how the beds change over time. Okay, so like I said earlier, I'll just be going over my PhD work a little as well, talking about plans mostly, but just a bit of context on what I do normally and how this is linked into my internship. So it's got a pretty wordy title, Genomic Analysis of Population Connectivity in the Scottish Marine Protected Areas Network to Inform Nature Conservation. So the original idea was to look into the population connectivity of priority marine features and see what this tells us about their management and if it could help inform us for future management and conservation needs. So the first species chosen was flame shells. As I described earlier, these are an incredibly important species and we just don't know enough about them. It'll be valuable for us to look into their population connectivity and learn more about how Scotland's populations are connected to one another. I'll also be looking at a second species, and razor clams were chosen as they are more important for economic reasons, so quite different to flame shells. So I'll just talk about where I'm at with these today. So I'm aware that not everyone here may be specialists in genetic connectivity, so I just thought I'd go over what genetic connectivity can tell us. So using genetics, we can find how closely related populations are. So if they are mixing or breeding together, and the more that populations are linked, then the larger the genetic pool, which is a positive thing, as this means populations are more resilient to change and more likely to recover to damage. And this means that we can also find out if populations are isolated, which is important for us to know in a management and conservation context, as such populations will not be as strong or resilient and will need more attention or care to conserve. So this will be valuable for us to know about our flame shell populations to find out how well connected the flame shell beds here in Scotland really are. So for the samples that we've collected, we've got 506 samples in total so far, samples from some of the protected area locations and some from outside of these locations. So we've also got Orkney to the north, which is a bit of a distance away from our western populations, and also some samples from the north coast of the Republic of Ireland. So this is interesting as flame shells are not protected in Ireland, so seeing how they're connected could be quite interesting for us. It's also interesting as it allows us to see how well connected our Scottish populations are with those from other countries, or if Scotland's populations are isolated and just mix them with one another. So of the 506 samples, we had 181 samples sent off to Florogenics for reduced representation whole genome sequencing, which is done via RAD sequencing. So in RAD sequencing, the whole genome is cut into fragments, which is, in this case, was done by single digest with the enzyme PSTI. Then a small portion of these fragments is sequenced, so we won't have to sequence the whole genome of every individual. Now I'm at the stage where I'm producing single nucleotide polymorphism, or SNP markers, from the sequence data. A SNP is a change to a single base pair in the genome, which we can then compare across individuals or across populations. So my goal is to identify a set of these markers, which I can then use to develop targeted genotype by sequencing assays, which will enable fast and cost-effective investigation of genetic connectivity and other genetic parameters across and within flame shell populations. By doing this, we can include all of the current flame shell samples that we have, and also include any samples which we might collect in the future, which hopefully we will if the pandemic allows it. So outcomes. The hopes for this project are to find out how well connected the Scottish flame shell beds are to one another, and with any other populations we get to include in our analyses. Such connectivity is important for understanding the threats of pressures such as fishing activity and climate change, and determining a population's ability to recover. So this knowledge will help us to understand how vulnerable our populations are, and as such will help to inform management decisions in the future. So how does this link with the internship? As well as just being studies focused on the same species, it'll be interesting to compare the results of my PhD work with that of the internship work. So if there are any genetic differences found in the beds, then it would be interesting to see if they correspond with any differences we found in the internship work. So that this could maybe hint at ecological differences driving variation in the genetics of populations. It also differs though, as the two can tell us different things. So the internship work tells us a little more about the whole ecology of the beds and understanding the interactions with other organisms, while the PhD research will look more at genetic differentiation and diversity and tell us more about the relationships between Scotland's flame shell beds and beyond, and future recovery. So both are hopefully something which can be used going into the future, with the internship methods hopefully being used to monitor changes to beds in the future, and the PhD generating markers which can then be used in the future for analysing more flame shell bed populations, should anyone wish to do any more research on the species. And then finally, we have the razor clam work. So in Scotland, we have 3 different species of razor clam. We have Entis entis, Entis arcuatus, and Entis siliqua. And these are shown in the image to the right. So you can visualize how these species are phenotypically different from one another. So collection was starting to go underway at the beginning of 2020, with some samples collected for me by Marine Scotland in their electrofishing trials. But then, of course, lockdown came and this sample collection was no longer possible. So I've had to reconsider things, and I'm now planning to travel around Scotland sampling individuals from the tidal area. So I'm planning to swab the DNA and re-release individuals after getting pictures, so the species can also be identified through our pictures. And then using RAD, I'll be looking at population connectivity, but also exploring species differentiation. As we can see, these species differ in visual traits, but we don't know how they are genetically related, and we don't know how closely genetically related they are. And so this will be an interesting thing to look into. So hopefully I have given you a good overview of both the internship work and my PhD work here. I'm relieved to have made it through because it's definitely the longest talk I've ever done. So thank you for listening to me, and a big thank you to my supervisors for these projects, to Victoria Pritchard at the Rivers and Lochs Institute at UHI, and to Lisa Camphausen and Flora Kent at NatureScot, and to everyone else who helped with these projects. And yeah, thank you for listening. And we've got some time for questions now if anyone has anything they would like to ask.
All right, well done, Lydia. That was tons of information in there, and it sounds like you you got a lot done in those 3 months with the internship, so well done. And it's really great to hear how it links in with your PhD. We've had a couple of questions submitted which we're going to work through in just a moment, so if anyone who is joining us on YouTube or Zoom, then please check, just submit your question in either the chat or the Q&A, and Lydia and I are going to work through those questions, and I can see a couple of others are coming in, so please submit them at any point. We are scheduled to end just before 2 o'clock. So Lydia, I'm going to ask a question, just to kick things off. So with the sampling that you've done for your PhD, you may have said this already, but are you sampling from the same locations where sampling was done for the work that you did on the internship? So you're able to, in a sense, almost look at some of the work that you did with your internship and the PhD because they were the same sampling location.
Hopefully, at the moment, we do have, I think, from two of the locations that I have for my PhD are the same as for the internship, and hopefully we will actually be able to get more from the other locations. Yeah, hopefully things can kind of get started up again with sampling. So yeah, it would be really good to actually, have from every location and see how they compare. So hopefully, yeah.
And I'm going to ask a real dummy question. What does sampling for your PhD mean with the flame shells? What are you going out? Are you doing these kind of, you mentioned the samples taken in the internship way, the cores or grabs, but what does it mean in your PhD when you're sampling these flame shell beds or where they are?
Yeah, I realised towards the talk, I was like, oh, I didn't even mention that at any point. So NatureScot have been collecting the samples for me, which actually started before I even started my PhD. There was samples being collected of flame shells. So it is taking a little bit of muscle and using that for the genetics. Yeah.
Okay, so someone's diving down and collecting just a little bit of tissue samples at all these different locations, and then you're doing the analysis of this.
Yeah.
Okay,
pretty much.
Excellent. Right, we're going to head into the questions that have been submitted by the audience, starting with Matt's question, asking, has carbon stable isotope analyses been performed on Scottish flame shells? They're aware of isotope mapping that has been used for oysters to determine their geographic location.
I think I've been asked something similar in the past about carbon within them, and I don't know much about that personally. I don't believe it is something that has been done, but it could be something of interest in the future, I guess.
Okay, great. Next question is from Katie, asking, were the samples from Loch Carron taken from the quite small area of the damaged bed or from a wider bed?
So there's the few kind of samples taken were dotted about the bed, and I don't actually know the area where the damage was, so I can't actually definitively say this is part of the damaged bed, so whether it falls in with the others or not, it's kind of hard to say because I don't know exactly where the damage was.
No, understood. Okay. Next question is from Lorito. At what level do you ID the species in the Loch study?
So species identification was done by other people, not me, but most of it was done down to species level, and if it couldn't be, I decided that I should cut off at the family level. So if the family share the same traits. So if I couldn't if it was identified to a point where I couldn't say the traits that were, present within that like group, then they weren't included in the study. So it was mostly to species level and some to more just family, but yeah.
All right, great. We are getting a whole variety of questions coming in now, so let's get to it. Next question is from Irene, asking, what are your thoughts on including environmental variables such as nutrient levels, proximity of fish farms and currents on the context of genetic comparisons?
Yeah, that would be really great to have those types of things also involved. I don't know. Oh, I've lost it now. It's scrolling away. I think it's maybe something more when you have the exact genes and stuff, you maybe look at how they could be influenced by these different factors. But yeah, I would definitely be interested in including them in any way that I could, to see how the environment might be affecting any differences between populations.
Mm-hmm. Yeah. Our next question is a bit more of a comment. It's from Claire, just highlighting that there's been work done at Aberystwyth University showing that Fraser clams that appeared morphologically as 3 different species were actually shown to be all the same species using genetic analysis. So that's quite an interesting link that might be interesting to follow up on if you weren't aware of this.
Yeah, that's a really good thing to point out. I'll have to look that up because that is like really important for what I'm wanting to look into as well.
Mm-hmm.
Yeah, so it'd be interesting to see if I find the same kind of thing between those 3 same species, if it is. And yeah, it's really interesting because they look so different. So
mm-hmm.
It'll be interesting to see if there is any, genetic differentiation, even if they seem to be the same species. There must be some differences in there, maybe.
All right. Our next question is from Catherine, asking, how many informative SNPs do you plan on to ideally use for future monitoring assay?
So for the flame shells, we're hopefully looking to identify a couple of hundred. So yeah, maybe around 200 informative SNPs that we can then use for the assay.
Great. I'm going to take a question that has come in through the chat. I'm not sure if Lydia, you spotted it, but I don't think it's been put into the Q&A. So it's from someone, Alexandra, asking, did you consider Bray-Curtis similarity index in your analyses? I don't even know what that is.
I think I did try it, but I would have to get it up now and see what the results were. But yeah, I think I did give that a try.
Mm-hmm.
I have it here. Let's see.
No worries, Alex. We spotted your question.
Yeah, so actually that is a part of the NMDS. Yeah, I knew I recognised the name.
And remind us, what is the NMDS?
So the non-metric multi-dimensional scaling. So that was the first figure where there was the colourful little groups, if that makes sense for you.
Yeah. And for anyone who wants to look back, this will be in the recording and they can look back themselves. We're going to move on to the next question, which came in from Graham, asking, might there be a difference in salinity between the 2 Korean sites that could have caused the observed difference in the 2 samples? Is that something that might cause a difference?
Yeah, it would be good to have those measurements of salinity. Maybe in the future that could be something that they consider, maybe getting some environmental variables such as salinity and different things like that to actually include in the analyses would be a good idea.
Mm-hmm. Yeah. Our next question is from Emily, which I think is a really interesting question about public databases. Are there any with pre-existing records that help inform your analyses? Was there anything out there already?
So I used the biological databases for the trait assigning. That was it for the public databases, I believe. Yeah, though I think the species identified in these samples is also public, I think, in a NatureScot report.
Yeah, okay, great. Our next question is from Matt, saying, thanks, Lydia. Some interesting differences in the faunal analyses. To what extent did you account for differences in the environmental parameters? So something we've already touched upon, but depth as well, current, speed, sediment type would normally be significant drivers of such changes.
Yeah, so I was able to look at depth and see if it was looking like depth was maybe influencing these results at all. Depth didn't seem to be a factor, but yeah, it would have been good to have some more environmental kind of variables that I could have included.
Mm-hmm. Just out of interest, how far down, what kind of depth can we find flame shells? Is that something that we even know how deep they are found?
It's normally between 5 to 13 meters, but it can be deeper or even shallower. But that's kind of your common.
Yeah, because I noticed the sampling that had happened off North Ireland, that was quite off the coast, so that must have been quite deep almost.
Yeah, now I'm thinking, what was the depth on them? That I'll have to look back and check what the depth is there.
Yeah. We have one more question that has been submitted from the audience. So if any of our audience who are still with us have something that they would really like to ask, please pop it in the Q&A chat, the Q&A box at the bottom of the screen, or if you're joining us on YouTube, you can ask a question on there as well. So we're going to go to Jonathan's question, which says, they noticed that some of the sites are in the same area as recently designated temporary MPAs in the inner sound of Skye for flapper skate. Do you think that this designation was influenced by the existence of the flame shell, or do you think or hope that the designation will help the populations of the flame shells?
Yeah, it could have been if the flame shells had been identified prior, which they should have been. Then yeah, I think that is an influencing factor for MPAs. They seem to generally be put in MPAs where they find flame shells, and yeah, I hope that it helps. I guess it's part of what we're looking at. Is the whole point, I guess, is kind of partly seeing, do we think it is helping? Which, yeah, I hope it is.
Yeah, well, as you recommended, doing constant some more monitoring and sampling will help determine that. Yeah. I've just noticed a question which I accidentally skipped over, which is from Hannah, saying, were there any samples you could have used from the Orkney flame shell population? So I think she's referring to actually the work in the internship. Was there any samples you could have used from the Orkney flame shell population?
So for the internship, I don't believe any kind of core or grab sample type sampling has been done. Maybe in the future. So if they do continue to do some of these, do collect more samples, they might include more locations as well, which I guess I would probably maybe also recommend to get more in there, find out what's happening, you know, in the broader scale at all the places.
Okay, I can't see that any more questions are coming in. So Lydia, we've got a bit of time. Do you have any other comments that you would like to say about this work? You know, you've got a bit of breathing time. If there's anything else that you're like, oh, I didn't have time to say.
I mean, I think I was talking so fast, I can't remember what I said. So if anyone else has any more questions, I'm like open to any emails, you know, send anything my way, that's fine. But yeah, just thank you for listening. Thank you for attending.
Yeah, thank you everyone for submitting questions as well. Yeah, and as Charlotte's just put into the chat, looking forward to the results of both the internship that will be coming out and your PhD too. So Lydia, best of luck with all the studies left, and hopefully you'll be able to get out and about and do some more sampling when permissions, when COVID restrictions permit this. Yeah, absolutely. Okay, so that just leaves me to say thank you very much, Lydia, for being today's MASTS webinar presenter.
Thank you.
If you're still with us right now, I would just like to highlight what MASTS webinar is happening next week. So this was with Douglas Spears from the University of Strathclyde, modelling zooplankton and fish and space and time and under climate change. So if this is a talk that you would like to watch and participate in the Q&A, then just use the same sign up link that you did for joining Lydia's talk today. And I would just like to highlight that we have a whole variety of talks coming up in rest of March, April, and May. So all the information and registration links for these webinars is provided on the MASTS website. So if there's anything here that catches your eye, then please head over to the website and sign up for any of these talks. They're all happening at 1 p.m. in the afternoon and are every Wednesday. So I hope you enjoyed today's MASTS webinar, and we look forward to seeing you soon.